Okay, so we'll go ahead and get started, everybody. So thank you for coming to Calvin's talk today. Just as a reminder, this session is being currently recorded. It is not currently being live streamed, but it will be uploaded to YouTube over the weekend for future viewing, just so everybody is aware that the session is being recorded and will be publicly available. So today we have Calvin Lam coming to us from the University of Nebraska to present some of his work on synthetic biology. He is currently an MD, PhD student, again, like I said, at the University of Nebraska Medical Center. He's an independent researcher in synthetic biology currently on the side, though. He first worked with CompuCell3D developing synthetic biology models in Dr. Leonardo Morset's lab at the University of Southern California, where he studied before Nebraska. His research focuses on biological self-organization, specifically on seeking to understand how small individual units, such as bees and cells, communicate with one another to yield emergent behaviors. He's currently working on synthetic circuit design for mammalian synthetic development and synthetic immunotherapy, with an ultimate goal of developing synthetic circuits that control immune cell behavior and therapy. So with that, I think I will let Calvin take over.
Great, thanks for the introduction. Today, I'm going to be giving an overview of my synthetic biology research and how computational modeling is very suited for being applied to this field in general. So first off, there we go, that's better. Cool. So first off, I just want to begin with a lot of background. What exactly is synthetic biology? So the field originally began in prokaryotic cells like bacteria. The whole goal of it was to think of it as an input-output system. Can you program bacteria so that when you give them an input, you get a specific output out? And the analogy that I like to draw to this is very similar to computer engineering. When we build computers, we start off with these component tiers. You have your CPU, you have your RAM, you have a bunch of other modules that do very specific functions in your computer. You integrate it all together into one piece. And usually what happens is that when you use it, you get a very controlled output. And this is the part that I really want to highlight here. You get a controlled output. You put in 1 + 1 into your computer, you'll get 2 repeatedly, barring, of course, you're unfortunate enough to get some sort of software glitch or faulty hardware. But again, the whole point is that the output's really controlled. We can think of cells in a very, very similar abstraction. Cells have very functional basic components. For example, your DNA encodes all the genes that you need in your body and more, for example, like long non-coding regions. This converts to RNA, and your RNA makes proteins that actually do the functional behaviors of your cells. Now, there's more modifications there, for example, like glycosylations, ubiquitination, etc. But the important part is when you take these components, jam them together, you integrate them into a cell, the cell can generate output. But the difference here between your computer and your cell is that the output is just whatever the cell natively outputs. For example, in hematopoietic cells, if I give them the chemokine CXCL12, the cells will migrate in response to that chemokine. I can't change that response. If I give them CXCL12, they'll migrate. I cannot rewire that output to a different behavior. So this was a challenge that honestly was a big challenge for the past few decades. That's why a lot of the work done was in prokaryotic systems like bacteria and so forth. However, in the last decade, we've actually started developing a lot of different synthetic biology tools that allow us to reprogram cells with a controlled input-output logic, especially in mammalian cells. This is the receptor class that I'm going to focus on for this talk. They're called synthetic juxtacrine receptors. And the reason for this is that they allow a cell to sense a juxtacrine ligand that you get to choose. You get to choose the ligand that this receptor binds to. And for the intracellular domain, you get to choose what gene gets controlled by your receptor. So effectively, you can think of these as like almost designer receptors. You can control what a cell senses and behaves using these types of tools. And as a disclaimer, I want to highlight that this is a very small overview of the tools that are being built in synthetic biology right now. There's a lot of tools being developed that can sense soluble ligands, etc., control different genes, both native and endogenous as well. So even at this very beginning of this talk, there's a lot of room for growth, because if we can do this type of signaling in silico, we can also expand it to all these other different receptors. So how these receptors work biologically is that they're usually based on a normal proteolytic receptor. For example, I'm going to talk about the SJRs in the context of the SynNotch receptor, which was the first generation receptor, the prototype. Second generation are called snipers, developed by Cole Roybal's lab at UCSF, but they use effectively the same design. The second generation is just humanized and slightly improved in functionality. So what we have here in this cartoon is that we have the extracellular ligand binding domain. And typically how this is achieved is by taking your synthetic receptor, and this part here can be a DNA that codes for an antibody, not an antibody, but the fragment variable domain of an antibody that binds a target protein. As a very common example, CD19 is a protein that people are develop CAR T cells against. The nanobody for that has exceptional affinity for CD19. It's very specific. You can imagine that if you want to target a cell that has CD19 on its surface, you can do a CD19 nanobody or antibody binding domain here, and this receptor will just bind CD19. That'll be the input that it senses. The next important part is that you have the intracellular domain. Now, what's being used right now here is usually transcription factors. These transcription factors can be native ones that the cells have, for example, like HMGB1, et cetera, or they can be orthogonal to a native eukaryotic system. For example, you can use bacterial transcription factors. You can use tetracycline-based transcription factors, which are bacteria native. Therefore, they shouldn't interact with if you put it into a human cell, shouldn't interact with the native system, except for the gene that's controlled by that you put in as well. You can program cells in a logical manner. With the extracellular ligand binding domain, you can have cells say, hey, if I put this receptor into a cell, if you send CD19 on another cell, I want you to express this gene as a result. We can have them express fluorescent reporters. Those are very common for reading the output, just seeing if your system works. For example, if this cell program receptor here, this binds CD19, this activates GFP. If the cell binds another cell that has CD19 on it, it should turn green because it expresses GFP now. Modularity comes in as a very powerful system because you can say this could be for cadherin expression that regulates adhesion, which will actually be the focus for today's talk. But you can expand this, and there's already been examples to expand this to causing apoptosis, changing the balance of stem cell factors, changing other transcription factors, and so forth. A lot of modularity here. The way these receptors work is by when they bind their domain, mechanistically, this causes a pull mechanism. So basically it stretches out the receptor and it exposes a protease cleavage site that's highlighted here. I want to highlight that this is the current belief mechanism of these systems, and the current data supports this model. But in the future, there could be changes to this. And the reason why I say this is because Dr. Roybal, who's developed the second generation receptors, recently gave a talk and told us that some of his receptors can sense a soluble ligand. So it's possible that the pull mechanism is just sensitive enough to a soluble ligand that exposes it. But in general, this is just the mechanism that we have and we can trust for now until more data comes to light that goes against it. When the protease cleaves this site, what happens is that you release this intracellular domain to transcription factor, and transcription factor can activate your gene of choice being an endogenous gene or an exogenous gene, depending on how you design the DNA binding domain that's close to your promoter for your gene. So with this, we have a lot of different control, a lot of modularity. We can kind of do a lot of different things with it. There's a lot of potential here, but like I mentioned, that the field is pretty new, especially for mammalian synthetic biology. These tools came up in the decades, but they give us the potential, or rather, they've given rise to several different sub-goals in the field. One of them is synthetic development. And the goal of the synthetic development subfield is to try to understand native developmental processes by building them. Here I've shown an example from an old simulation. The goal here is to build a lateral inhibition circuit, which generates a checkerboard pattern. So you can imagine like one red cell surrounded by green cells. Red cells don't touch other red cells, they just have green cells in between. Excuse me. These are native patterns that occur in a lot of developmental processes. For example, the hair on your arm. You ever wonder how they're spaced out so neatly? They're spaced out because of checkerboard patterning, similar mechanisms to this patterning here. In your inner ear, you also have checkerboard patternings here. That's how you get cochlear cells to form pairs that enter at different spikes, and they're not randomly formed. They're actually formed in a very organized manner. From a therapeutic perspective, there's a lot of growth potential in this field as well. If we can program cells to generate the patterns and the tissues that we want, What's to stop us from building the organs that we want or tissues that function just like organs that we want? Can we use this as a way to address the need for organ donors? Can we build organs in the lab and transplant them into patients to cure diseases? The part of this that's perhaps solidified the most right now is synthetic immunotherapy. I've earlier mentioned that there's a CAR T cell development, very popular therapeutic strategy. You can basically program a T cell in response to a antigen, kill the cell with the antigen. But that's kind of limited. You just have one receptor there. It's a if A, then B type logic. But with these synthetic receptors, if you couple them with chimeric antigen receptors, you can say if A and B, then kill the cell. But if there's a C antigen here, don't kill the cell. Commit suicide instead, so you don't actually kill off healthy tissue. There's a lot of modularity enabled by these systems. I've shown that I've talked about how you can combine different receptors together. You can combine synthetic receptors with CAR T cell receptors and so forth. Problem is that this creates a lot of permutations. There's a lot of circuits you can make, and it's not easy to predict the results. From this, for example, you can program a cell of actually oscillatory circuits. In the single cell level, you can cells activate a gene, the gene inhibits itself, and you can actually see gene expression of that protein go up, oscillate down, up, oscillate down, up, oscillate down. But what if you make it dependent on its environment? What if it depends on a ligand from your neighbor? So if the oscillation actually depends on what the ligands are expressed by its neighbor, and if the neighbor is also oscillating with that ligand too, the systems are very nonlinear. It becomes really complicated to predict the outcome of once we're in a multicellular context. This is where computational models come in. Because we can program cells in silico with a similar program and basically just test and see what we get out of it. We can use it to parameterize against a test set of structures. We can get information out of that. And then we can use that to generate predictions about our system if we made different modifications. So just as a simple example here, an in vitro example, we take a cell, gray cell here. We program them with a synthetic juxtacrine receptor that senses orange ligand on orange cells. Cool. When they sense this, they basically release the red intracellular domain here, which is our transcription factor, and it activates target gene expression. Let's say target gene just makes the cells blue. So from this type of logic, if these cells contact one another, they're able to continue signaling. This cell here is going to turn blue over time. In silico, we can perform the same logical design. We can have orange cells, we can have gray cells. When these cells touch one another, basically they're sharing surface area, juxtacrine signaling. We can calculate how much signal they get based on that contact area. We can activate the expression of a target gene, and in this case, we can calculate as a number of proteins in the cell, so forth. Once they accumulate enough proteins that turn them blue, we'll just make the cell turn blue, and that's our in silico analog to our biological system. So I'm gonna have a little brief break here for questions right now. That covers effectively the background part of this talk. For the next part of the talk, I'm gonna jump into the actual computational part and then provide an example that we'll walk through. If we have no questions, I will go ahead and proceed on to the next part of the talk. Cool. All right, cool. Oops. So I'm gonna walk through an example, a 3-layered structure. I'm gonna describe how I use CompuCell3D for synthetic biology. Let's say in theory, what we want to design is we want to design this 3-layered structure here. There's green cells in the middle, red cells that surround it, and blue cells around on the periphery. In silico, this is a result that I'm showing here, and this is a known biological result, showing that computational models can indeed be powerful tools for studying synthetic biology systems. So if you guys want to right now have access to the code, you can go ahead and open up in TWEdit++. Go ahead and open up the 3-layer program, or rather the 3-layer one. And we can load up the 3 codes. We have an XML file that contains part of the code. We have a PySteppable file that contains our Python steppables. And we have a Python code for initiating our steppables, if you guys want to do that right now.
I will go grab the links for where you can download these.
Sweet, thank you. So I do want to mention, just as a quick disclaimer for these codes, they demos, Unfortunately, it takes around 2 to 3 hours. So at the end of this talk, I'm going to have a Dropbox link to the output lattice files. And for the biologists in the room, the lattice files, you can think of them exactly like the Z-stacks that you took on a confocal imaging or on a microscope. You can basically replay it. You can look through the different layers from different dimensions. You can basically reconstruct and see your structure in 3D at any time. Sweet. Thanks, Hayden. Cool. And then we'll have time to look over the code again at the end too. No problems. All right. So let's figure out how we can build our 3-layered structure, our synthetic development structure here. First thing that we need to do is we need to develop a developmental trajectory. Basically, we need a sequence of steps where we can program our cells and get them to logically form what we want. So there's many ways to build a 3-layer structure. I'm going to say that right now. There's tons of different ways. This is the one that we'll focus on in our demo. So I want to start with a mixture of blue and gray cells. These cells are in vitro. They're blue because they have a blue fluorescent reporter expression. Gray cells don't have a fluorescent reporter expression, so they just stay nice and gray. I want these blue cells to signal to these gray cells. So these gray cells should be able to sense ligand on the blue cells, and that should turn them green. So they express a green fluorescent reporter. In this case here, our reporters that double as our ligand, so it's a GFP ligand. And they should also express high levels of an adhesion protein, which cause them to form a little cluster at the center of the structure. Now, to get this red layer, what I propose is that we have these green cells with their GFP ligand on their surface signal back to the blue cells and turn these blue cells outside red, so they express some adhesion protein, not as much as our green cells, so they can't compete for the core, but they wrap tightly around to form our layer. And I also want these cells to turn red. So when these green cells signal to them, I want these cells to be a little bit adhesive and express a red fluorescent reporter, for example. And that should form our 3-layered structure in theory. Let's walk through the code for it. So this is the signaling network that I'm going to refer back a lot to. I know this is a lot of information to take in. So what I'm going to do is I'm going to reference this signaling network as much as I can throughout the subsequent slides and how they relate to the code. This is our steps that we'll use. So we have our blue and gray cells here. That's our initial starting population. We have red and green cells here, which will be our quote unquote activated state respectively for each of these different cell types. For step 1, what I want is these blue cells with their triangle ligand on them to activate the synthetic juxtacrine receptor on gray cells that senses this triangle ligand. This should drive the expression of high amounts of the E-cadherin protein and GFP ligand. E-cadherin is a cadherin that makes these cells adhesive to effectively other E-cadherin positives. Once we have this triggered, these gray cells, they should turn green. They express high E-cadherin. The way I've depicted this here is that they have 2 E-cadherins on their surface when they're green. Plus, they should express GFP ligand, which is here on the surface of these cells. This GFP ligand should signal back to blue cells, which have an anti-GFP ligand synthetic juxtacrine receptor on their surface, and that should drive the expression of low E-cadherin levels and a red fluorescent reporter protein. After the cell accumulates enough of these cadherins and red fluorescent reporter, or red fluorescent reporters are readout here, these cells should turn red and they have some E-cadherin on their surface. This is the roadmap that we will refer to. And I'll walk through now the code and how we can build each of these parts piece by piece in CompuCell 3D. First step, we need cells. So how do we do that in CompuCell 3D? Well, fortunately, it's a pretty straightforward process. If you guys are able to open it up, it's in the XML portion of the code. We have a plugin here for cell type. We can basically specify what our cell type is here, and we can specify a type name too. So for example, type ID1 refers to these gray cells here, and their type name is Y. Type 2 is G here. Type 3 is our blue cells here. Type name B. 4 red cells R. You can probably guess that when I made this code, I named it in the manner that I tried to match the type name to the color. After having gained more experience in working with CompuCell 3D and coding in general, my recommendation is actually don't follow this type of naming. For example, I kept it similar in another project where I look at inhibitory circuits. And when half your cells end up being 2 colors that are very similar, like green and cyan, you want to recolor so the contrast actually looks way better. And the naming then becomes really confusing because type B, which you naturally associate with blue cells, could be representing green instead for some reason. So just as a little lesson that I've learned throughout my time, when you choose your type name, choose it in a way that'll work with any color. Don't name it after the color. So now that we have our cells built, we can start adding physical properties to the cell. So one of the first things that we can do is we can add the adhesion to the different cells. And the reason why I'm adding the adhesion now is because it's still in the XML file of the code. So we have a plugin named contact here. And basically we can specify how different cells adhere to one another. So based on our hierarchy of expression, green cells, they should have a lot of E-cadherin on their surface because we target expression of high E-cadherin levels. Red cells, they have lower levels of E-cadherin. These blue and gray cells, they should have less. So if we look at our hierarchy, for example, green to green, G to G here, see I'm referring to it as color even though I shouldn't, G to G, 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 the energy here is 20. The way energy works, the way adhesion works in CompuCell3D is that the lower the value is, the more adhesive the cells are to one another. So if you look in this hierarchy ranking here, 20 is our lowest value, representing that these high E-cadherin cells should be most adhesive to one another. Red cells should be more adhesive to green cells than other red cells. That's why we have a value of 35 here. And the red cells in general, they're adhesive, but not too adhesive. We'll put them at around 40-ish. I want to highlight that these values here were derived from a different way. They were parameterized based off flow cytometry data. And ideally, the best way to actually develop your adhesion matrix is if you have flow cytometry data. For example, for these cells, you can stain for the levels of E-cadherin, run them under a flow cytometer. You can see the actual expression levels based on their induction. And that's the better way to do it because you can see the leakiness of E-cadherin expression. For example, in the actual in vitro data for these gray cells here, they had leaky E-cadherin expression. That's why I've left these gray to gray cells, Y to Y, slightly adhesive to one another. So I had in vitro data to parameterize this off of to predict and test other structures. But if you don't have in vitro data, the best way is to build a hierarchy based off logical deduction of which cells are most adhesive to which cells. And I will say this, the important part is having the hierarchy overall correct. Honestly, if I ran this at 49 or I made these gray cells and blue cells completely non-adhesive to one another, I'm reasonably confident that I would still get the same three-layered structure out of this prediction. It's just that if you're looking at a biological system, it's not perfect. You have leakiness of background gene expression, you have leaky expression. And in this case here, when I looked at the flow cytometry data for these gray cells, they already had some leaky E-cadherin expression in their system. Blue cells, they had much less leakiness, probably because they had a low E-cadherin gene rather than high E-cadherin gene. So we have cells, we've added adhesion to them, and we've defined a hierarchy that helps us understand why our cells should behave the way they do. So next, I'm gonna walk through other physical properties that we need to define for ourselves. So right now, I have an initialization here, and this falls under the the initialization for CompuCell3D. You need to initialize some parameters for your cells, otherwise you don't know what to do for the initialization. So for our cells defined in our cell list, I define these parameters here. So for the cells that I focus on for this study, these cells are L929 mouse fibroblasts. These are naturally adherent cells. When you plate them, for example, in a T25, they form these nice little spindly cells. They look like 293Ts, honestly, if you're familiar with PEC cells. But for a three-layer formation, this was performed under ultra-low adhesion conditions. So this was a U-bottom well where the cells basically prefer to adhere to each other much more than they would adhere to the actual plastic of the plate. So I want these cells to have a spherical morphology because under these conditions, they're effectively little suspended spheres. So I defined a cell dictionary here called random. And the reason for this is that it basically generates a radius for each cell that we initialize. And this radius is drawn from a Gaussian distribution with a defined average. I haven't shown these are fixed variables here that you can define the code. In this case here, I define it as a 3 and 0.1. And basically what this does, it'll initialize your cells. They have a radius that's assigned to them, and they'll try to target that radius at a spherical morphology through their target surface 4 pi r squared and their volume 4/3 pi r cubed. So this is just a rudimentary way to get our cells to have a defined radius for each one of them. And they'll try to target a spherical morphology by changing their surface area and volume to match a spherical morphology. This is a useful piece of code to have because if you guys have done cell culture and you looked at your cells under trypan blue and under a scope, for example, the morphology is not always perfectly uniform. They could always be spherical, but you'll see that some cells are really large, some cells are small. And it's nice to have that in your in your computational system as well. As another part, I haven't shown mitosis here. I actually removed it out of these demo codes to keep it simple. If you add mitosis, you need to define a growth model for them. So you can have your cells simply grow and they can double and then you can divide, form two cells. The problem with that in your in silico system is if all your cells have the same initial volume that they target with or start with, or same volume, and they grow at the same rate, your mitosis between all your cells is going to be synchronized and all your cells will try to divide at the same time. And in a biological perspective, that's not really representative of all. Your cells are probably in different states of the cell cycle. The mitosis rate should be relatively constant throughout your experiment rather than synchronized to one specific time point. So for these parameters here, lambda surface and lambda volume, they basically tell us how well can the cells follow this these surface area and volume, target surface area and target volume constraints. If this value here, I've kept as 2.5 for both of them for simplicity, you can change these constants depending on your cell morphology. For example, there's good in vitro evidence that when these cells become adhesive, they actually deform really well. They stick to one another because they're expressing cadherent molecules and they like to flatten and adhere to one another. For this simulation, I'm going to keep it at 2.5 to keep it simple. But you can imagine for a cell that's actually adhesive and you're interested in modeling that deformation, you would probably lower these constraints a little bit, let your cells be a little bit more flexible when they stick together. If these values are too high, your system will become stiff. Your cells kind of get stuck because this effectively makes it very hard for your cells to make pixel copy attempts, which are how cells try to move in CompuCell 3D. I've also defined a cell dictionary here called points, and I've set this here to 0 for now. This point here is effectively going to represent our green fluorescent reporter, our GFP ligand in these cells, our red fluorescent reporter here, because these are the readout for whether these are our readouts in vitro for whether the cell is at a high E-cadherin expressing state or a low E-cadherin expression state or a baseline state here. So I started the cells here with their dictionary at 0 because at time point 0, I only have blue and gray cells. Ideally, they should have very low levels of red fluorescent protein E-cadherin. They should have low levels of GFP ligand, ideally. I also want to point out that you can redefine these parameters in the subsequent part when you actually run the simulation. I mentioned that adhesive cells can become more adhesive. So in this code here, I specified cell type 1, which are gray cells, which are here. You can imagine for this code for green cells, if you want your green cell morphology to actually deform more, you can lower these constraints to 1.5, 1, etc., and so forth. That way your green cells are allowed to deform. That could be a way to get that process there. I also want to specify here this cell.fucked_ample, which is code that codes for motility of the cells. So when we developed this model, we initially started with a constant motility just to keep it simple. But later on, as we talked with other reviewers and talked with both reviewers and other labs, a really common thing that's done in these models is that you actually have your cells change your motility based on its environment as the simulation runs. So instead, we chose to redefine our motility as a baseline called this fixed number basal here that can define a scaling factor here that defines how well your adhesion attenuates your motility. And the whole idea behind this code here is that when your cells are in a very adhesive environment, for example, you have a green cell surrounded by a bunch of green cells, that cell should not really move a lot. It's stuck to its neighboring green cells. It's kind of stuck there. But if you got a green cell and it's next to a lot of blue cells, that green cell should still be able to move because the blue cells are not very adhesive. And how this works is this other portion of code that I'll show right now here. So I'll just run through this as an example part of this. So this is the motility for gray cells. Here in this line, you can see CSAY plus, Y to Y times common CSAY. CSAY is our common surface area to gray cells here. This is the Y to Y value. This is our gray to gray adhesion value here. And the idea then is if your cell is surrounded by a lot of gray cells, they should be pretty mobile in general. But let's say here, let's move to this part here. Y to G. For example, let's envision the gray cells are really adhesive. They're adhesive to green cells. This value here should decrease because the lower the value here is based on our adhesion matrix, that should be lower because they're more adhesive to one another. And correspondingly, multiply that by the surface area that the cells share with other green cells. So our focal cell here is adhesive, shares a lot of surface area with green cells, and this value here is naturally low because cells are adhesive. You're basically decreasing the motility overall of that cell because it's surrounded by a lot of high adhesive cells. These values are pulled from this neighbor code here, where you basically pull information from the neighboring cells. So common surface area Y here is calculated from here and here at each time step. For each time point, the cell senses how much common surface area that it shares with the neighboring gray cells, totals that, and they can use the feedback into motility. These values are fixed from your adhesion matrix. And you can see here, there's a little hint of other stuff. There's PTSY, PTSG. These are values that I'm going to talk about a little bit later on. These are values that are important for the signaling aspect of our model. So we defined the physical properties, the basic physical properties that we need for our in silico cells. They've got a physical shape. They like to be spherical. They have motility defined. They like to move about. Their motility changes on their environment depending on whether they're near other adhesive cells or not. Now we can finally start building our signaling network. So the first piece of code that I'm going to focus on goes back to what I just showed as a little teaser, is we need our cells to sense our neighbors, and we need to be able to program cells so that they have ligands as well. We haven't added ligand expression to our cell yet. So I want to highlight for this line here, points, PTSY stands for points Y, which is our points from our gray cells. If you recall from our signaling network, which I'll show here, gray cells don't have any ligand on their surface. At any time point, they should deliver 0 points to their cells. And as another coding alternative, you can actually delete this line completely from the code. It wouldn't make a difference here because gray cells have no ligands. In contrast, our green cells do have ligand on our surface. Our cell dot type 2, and the neighbor codes refer to neighbor of type 2. They actually have GFP ligand here on their surface that signals to these blue cells, but that's an inducible ligand. It depends on how well the cell is receiving signal from these two other cells through that triangle ligand here. So that's why when we calculate the points G, PTSG, you take the common surface area that this cell shares with points G, this is the delivery part here. So PTS, I'm getting ahead of myself. These cells signal back to blue cells. This is the points from green neighbors that these blue or red cells sense. Take the common surface area that these cells share with green cells here, and you multiply it by the neighbor dictionary, because the green ligand here is what we're tracking through our points dictionary here. When you normalize that to the neighbor surface area, so we're basically taking the total ligand over the cell, divide by the surface area to get the ligand density, then we multiply back by the surface area that this cell shares with this cell. So one assumption of the model that we're looking at here is that we're assuming that the receptor for these green ligands are in excess in the cells. In the in vitro experiments that these reference to, we had no evidence that the receptors were a limiting step, but it's absolutely possible to model this as well in a model as I described in my original manuscript. And I can talk about that more if we have questions about that too. For our blue and red cells, they have ligands that deliver to both gray and green cells. So for our blue cells, they have points B, PTSB, but note that instead of referring to a dictionary, it just refers to this variable here called conexp-SCF. The naming for that is a long story for another day. But this is a constitutively expressed protein. It's not induced. We don't expect the expression level to change on these cells. For example, if you program these cells in vitro, you could put them under a CMV promoter. So it constitutively and strongly expresses this triangle ligand. So no need to refer to a dictionary here. For the sake of our model, we'll keep it simple. We'll use a constant variable here. But just like with our green cell, what we do is we take our total level of ligand here, conexp-SCF, we divide it by the surface area of our neighbor here to get our ligand density on the cell surface. Then we multiply that by the common surface area that's going to share with the green cells, etc. And that's going to give us the amount of actual ligand that the cell sees at a given time. So the signaling strength is actually very strongly contact dependent. A green cell that barely sees a little bit of blue cell is going to it's gonna, but a green cell that's surrounded by a ton of different blue cells, it's gonna receive a lot of signal that's gonna strongly drive the expression of the GFP ligand at high EC adherent levels. Red cells, they express a triangle ligand, but they don't actually express an inducible ligand on them. That's why they follow the same logical code as your blue cells here. So with this set of code here, we've defined the ligand part of our cells. We defined the ligand on our green cells, on our blue and our red cells, and we defined how these points can be sensed by different cells, different neighbors. So now that we have cells that can sense ligand, now we need to make them respond to that ligand. So how do we do that? Well, in this section here where the cells change reporter as a result of signaling changes, I call a reporter here because in vitro our true readout is that our proxy readout is actually the fluorescent reporter level that's conjugate, that's linked to the expression of the adhesion protein. In this first line here, we have if the cells are of type 1 or green, they should change their expression level of the GFP ligand and EC adherent based on this equation here. This is a sigmoid equation here that's mathematically equivalent to the Hill function that's very commonly used for modeling these types of systems. When I designed this model, I kept it to the sigmoid equation because in my opinion, it's more intuitive for someone starting out to understand this equation. Basically, you have a sigmoid equation here with these parameters here. Most of them you can keep at 1. For example, alpha YG can be kept at 1. But the most important ones are literally these two parameters, beta YG, 3 parameters, beta YG, epsilon YG, and kappa YG. And they just control how well your signaling occurs. Beta YG, this is a math.exp as an exponential function. So you can balance really quickly in your head what happens if your points from your red cell and your points from blue cell at a given time are ligand. Let's say we receive a lot of points. This number is really high. It exceeds our beta YG value. What happens here is this is an exponential to a very negative number. That's approximately 0. You get very strong signaling. It's 1 over alpha YG. Alpha YG in this simulation is 1. So the change in the protein level is marked as 1 in this case here. We're excluding the degradation term for now. However, if this beta YG is super high, let's say to infinite, then this exponential here is going to be e to a positive infinity, which is going to be a massively large number here. So this 1 divided by positive infinity, that's effectively 0. So you'll only get decay here. You don't add protein to your system. So it's a really simple way to kind of understand how exactly this works. And it's intuitive because you can quickly do the math in your head, write out the equation, do the math in your head. If our cells receive this many points, do they actually make protein or do they lose protein as a result? Is there enough points to actually induce protein expression? Cat 1 over kappa here times cell.dig point 0. This is our protein level at any given time. That's why we assigned this at the very start of the simulation. There's 0 here. We started these, if you recall from our initialization code, we started at 0 reporter level here, 0 protein here. So this is just standard inverse decay rate that's used in a lot of biological models. We kept this part simple here. So at any given time point, what we have is we have effectively a math equation here that tells us our synthesis rate based on how much signal that cell is receiving from neighbors that can signal to it, and how much protein is being lost because of standard degradation. After we calculate this point here, that generates a value for this variable here called DTRES. Then the dictionary, which actually tracks our total protein level at a given time, updates according to that change. So for example, if you take a green cell, you isolate it, it could have a lot of GFP ligand and EC adherent at the beginning, but without the triangle ligand to keep that expression level up, that protein level is going to drop. It's going to have 0 here. This is going to take over. It's just going to keep decaying, and that cell is going to drop back to its gray state. The exact same logic applies here for our blue and red cells as well. Blue cells type 3, red cells type 4. They receive signal from points gray, and I'm going to talk about this in just a little bit. Sorry, not points gray. They receive signal from green cells, PTSG. They're controlled by a similar equation as well because this green GFP ligand activates the expression of RFP and low EC adherent levels. It decays here as well according to the similar decay model, but note that the parameters that I defined here, these variables, are separate. Numerically, I've kept them the same. Both of them are set to 25,000 in these simulations here. But from a realistic biological perspective, when you transduce cells, the Number of transgenes you put in will change depending on the transfection, depending on the single cell clone you select, and so forth. Not all transfected cells are equal, so these variables can easily differ between one another and even within the same cell type. That's why for these type of experiments, it's important that you can grow up cells that behave very similar to one another. And common way that people do this is you take your cells, you induce the expression of your fluorescent reporter in them, you sort for single cells, and you grow up a single cell clone that will behave very consistently throughout your experiment. Just like with the gray and green cells, after we calculate the change in protein at a given time step, we update that so that the total reflects that change as well. And I just talked about that. So going back to that little piece that I stumbled on, or rather wanted to talk about, note that we have a points Y here. I was revising the slides yesterday and I caught this. Now, I kept this here, it's actually an artifact that I kept from previous simulations because in the past I've tested simulations where the gray cells can actually have a ligand and can signal back and forth. So in this developmental trajectory and signaling network that we specified, gray cells should not deliver any points. They don't have any ligands on their surface. Green cells are the ones that do because they have GFP ligand. But recall earlier that I defined this points Y to always be plus or minus 0, that we could have actually deleted it. So for each time step here, we're actually just adding a 0 over and over into this formula here. So it actually doesn't change our results at all. This is one of the things that I want to highlight is that when you make template code, this is something I used to do in the past, when you make a template code, it's really important that you do QA, you check over your code, especially when you use it to make a new simulation. You should still line by line look over all your different lines, make sure everything's correct. And especially after you run all your simulations, you get your results. My personal policy is look over the code one more time, make sure that it actually models what you're looking for. Make sure you don't make a mistake where you actually accidentally included something that you did for this other model here and it's left over here. Very common mistake that I've done in the past before. Funny story in the paper that where we published this model, was like 4 months before that, I actually caught a mistake that I made because I used a template code and I didn't fix the template code correctly. So all my simulations were wrong, but they gave the correct result because the error was actually very minor numerically, but the code itself was incorrect. So I actually had to rerun and redo all those experiments. Nice part here, it's computational. You just rerun it, and typically it doesn't take too much time, especially if you have a really good computational system. So there we go. So with this, we've actually defined our signaling system. In the previous one, we defined the ligands expressed on our cells. Now we defined the signaling network. Now we can have cells actually sense and respond appropriately with these ligands on their surface. So now that we can have cells that can respond to this signaling changes, the next step is we need to make them actually change the state. Recall that in our demo, we defined these green cells already to have high EK adherent level, red cells to have low EK adherent level. Green cells are just really adhesive to one another, red cells are not as adhesive. So since we already defined those 4 cell types, all we really need to do now is have these cells transition to their quote unquote activated or adhesive form. So in this block of code here that I have in the Python file, if the cell is type 1, effectively these cells here, if the number of points that they have is greater than a threshold that we define, we'll change them to adhesive. If the cells are already type 2, they're already adhesive, but if they lose enough signaling and their protein level falls under that threshold, same logic holds here for these blue cells and these red cells as well. If these blue cells gain enough low EK adherent RFP levels, we'll promote them, we'll make them activated, we'll make them adhesive. I want to note here that this is a very simple way to model these state changes. It's a discrete model, it's not continuous. And from a biological system, most systems are continuous. EK adherent level when you do slowly goes up. It doesn't just jump from non-adhesive to adhesive. So this is a limitation of the model here. But I want to highlight that when I test this actual simplification, it doesn't really change the result from what I see. And that highlights that the model is just, sorry, not the model, but actually the 3-layer structure is likely very robust to this assumption here. If you want a system where that has to be more continuous, you can build that as well. Contact plugin that I use in the XML file is a simple way to define adhesion. But if I recall correctly, there's a way you can define the level of adhesion molecules on each cell. There's a plugin that does that. And that's something you can do. It does add caveat, of course, it's makes your simulations a lot more complicated, and it adds a lot more parameters that you're going to have to figure out. And for biological systems, for computational modeling, my policy is you keep it simple. You need to capture the phenomenon that you're modeling, but you want to keep it simple enough that it doesn't have too many parameters and effectively over models your system. You don't want to overfit your system, which is a problem. Very common problem in modeling, actually. Cool. We've got our signaling network defined. We've got our physical properties defined. We have one last piece that we need to add before we can actually start running our simulations. We need our initial condition. We've defined all our cells, we've programmed them, but we need to actually put them together, make them signal to one another, sense one another, and actually form our three-layer structure. So this is back in our XML file. And I've used a steppable here called blob initializer. So what this does is at the center of our 100 by 100 by 100 lattice that I specified in the plot section of the XML file, we have a little spheroid of cells that initialize a mixture of blue and gray cells. You'll notice here that I've actually specified 3 regions and they're concentric to one another. The radius of our first blob is 20, second is 16, and the least one is 11. So we have 3 concentric blobs. You can use just one region here, for example, here to here, and that's enough to actually get your enough to get your initial mixture of cells. But when I ran these simulations, one of the things that I wanted to improve upon was actually how close it was to the in vitro ratio. Again, this was the simulation that I was trying to use as a parameterization one to build all my other simulations on. I wanted it to be as close as possible to 200 blue cells and 40 gray cells. And I find that by doing this concentric initiation instead, it actually got me much more closer than just using one region to initialize. That was a lot of talking. Let's take a look at our in vitro output and we'll see how well it matches against our developmental trajectory. So let me see if, can I play this movie here? I have to press it. There we go. So this is the in vitro counterpart that's known to exist, and we can see how our structure looks overall. So when you have a mixture of blue and gray cells, cell signals to one another, you can see on the merged color field here, the cells turn green, they form a nice little core, and the red cells start forming around them at the end. This is another formation with a different ratio of cells. I want to note here that this is 160 blue cells and 80 gray cells instead. Forms a similar 3-layer structure here. So this is a very robust formation, depending on robust formation, even when you change the number of cell types or cell ratio. And here's the Z stack that they've recreated. This is the work of Satoshi Toda during his time and Leonardo's time at Wendell Lim's lab. This is their work. So I'll run through now the central symmetric 3-layer structure that I get from the in silico analog of this. There we go. So blob initialized, we have cells activate, they start organizing, they signal to one another, and we get a 3-layer structure formation in silico as well. So now that we have our system initially parameterized, again, I've left out biological parts like mitosis, their cell growth. If you do the in vitro counterpart with a few cells, you can see that over the course of like 72 hours, the cells actually double, they get bigger and bigger, your 3-layer structure gets bigger overall. I've left out mitosis here for simplicity, but we can start using this parameterization because we match pretty well. We can use it to make predictions. So I showed earlier the in vitro 160 blue cells and 80 gray cells. The end result is this structure here. In silico, we get a similar result. This is our end structure here at a similar in silico to real-time time point. And the way I've developed this is I manually calibrated 1,000 Monte Carlo steps to effectively 1,000 Monte Carlo steps to 1 hour of real time. And by using this time point to define our parameters, we get a similar development at these end times. Another example here, when we work at different cadherins. So this is not using E cadherin at all. This is using P cadherin and N cadherin. So the green cells express, it was either P cadherin or N cadherin, I can't really remember. But the idea behind these cadherins is that they're not adhesive at all to one another. In fact, they really don't like to adhere. So what happens is that you actually stratify your structure. You get a bunch of adherent green cells that stick to one another, and these red cells don't like to stick to green cells actually. They like to stick to each other more. So what happens is that you form these cross sections here with these poles of red cells, inactivated blue cells. In vitro, we get a really similar structure as well. And that was basically these developmental structures that we worked on at the very beginning. Since then, there's a lot of things that we can do with these different types of circuits. That signaling network there forms the foundation for a lot of circuits that I've looked at in the future. One of the things that we can look at is adding in temporal control. These juxtacrine receptors you need to have the signaling ligand present in order to actually get the result. However, can we make them less sensitive to that? Can we add more temporal control? One example that I proposed mathematically and in silico is adding a transcription factor as an amplifier. So this is an amplifier that you would draw, like for example, in electrical engineering. The synthetic juxtacrine receptor, when it senses these orange ligands, expresses another transcription factor. This transcription factor then activates your target gene. So it's kind of like an amplification, because as long as you have enough of this present, you'll still get target gene expression, and you can actually expand how long the cell remains activated, even when it loses signaling to the neighbor. You add complexity this way. For example, we can get a 4-layered structure, as I've highlighted here. And with these different levels of temporal control, you can get even striped multipole layers compared to the multipole structure we just saw. We can even add different layers here, like orange cells here, red cells here, pink cells, 3-layered poles, as opposed to the 2-layered pole that we saw of just red cells and blue cells. More different circuits that can be explored, a lot more different uses for these amplifiers as well. We can also model inhibitory circuits as well. I've only talked about the synthetic juxtacrine receptor activating gene expression right now. We can have it actually express an inhibitory transcription factor or a transcriptional repressor. For example, Gal4 crab is a transcription factor that's been used with these SJRs, these synthetic receptors, and they've been shown capable of repressing gene expression. So what happens if we use an inhibitory gene circuit that also combines activation like E-cadherin? This is a circuit that's also published in Satoshi Toda's work. If you want to take a look at that, it's by Satoshi Toda and Wendell Lim. They've published all the in vitro work on this stuff here. That's how I actually got my training set of data to predict all the air structures. But from a uniform population of blue cells, what you can get are these weird 2-layered structures that have blue cells on the periphery. Inside, you kind of have a mixture of red cells, blue cells, cyan cells. Cyan cells are the ones that are intermediate. So that was all the synthetic development portion. We used it to build structures. Cool. These structures are simple right now because the field is just beginning, but you can imagine that we can create more complex programs. Can we advance these structures to a more actual functional tissue-like level? The other field that's using, not using CC3D, but using synthetic biology is synthetic immunotherapy. Can we program these immune cells with smart, complex programs, have them selectively target and kill cancer cells? For example, in this graphical abstract that I've shown here, this synthetic biology CAR T cell requires all 3 antigens before it actually can kill a cancer cell. Cells that have 1 antigen or 2 antigens here, but not all 3 antigens, are spared by these types. And an example that I want to highlight here is that I have a CAR T cell here that's programmed with a synthetic juxtacrine receptor that senses a yellow antigen. When it senses the yellow antigen, it activates expression of an anti-red antigen. That's why I called it ARA-CAR. And then there's the anti-red antigen CAR. In silico, we can create actually like little pseudo tumor setups. We can see a mixture of a surrounding, and these tumor cells can have different antigen expressions on them. In this example here, I have surrounding S cells that are orange. These are healthy cells. They don't express any of the antigens that we want to target here. We have dual antigen tumor cells that have yellow and red antigen. We have a tumor cells that have only red antigen. And of course, we have our SB CAR T cell, because I call them synthetic. These synthetic biology CAR T cells, they can activate off these dual antigen tumor cells and kill both these red cells and these pink cells. Basically, they can eliminate a heterogeneous antigen expression tumor, which is a very, very common challenge right now for immunotherapy and regular CAR T cells. If they don't have that antigen, they don't get killed. But this way, you can target both. You can actually kill tumor cells with both antigens and single antigen positive cells as well. Again, I want to emphasize there's a lot of room for expansion, especially in synthetic biology and with the computational model too. I just want to highlight these are 4 different synthetic receptors that sense soluble ligands. So unlike our synthetic juxtacrine receptors, these are built to sense only soluble ligands, but they're also perfectly modular as well. These are nanobody or antibody binding domains. You can sense a ligand of choice. This is a transcription factor that controls your gene of choice. So one of the things that we can do with this model down the line is we can expand it for our soluble molecules. Then that lets us ask the question, can we build signaling networks that utilize both soluble ligands and juxtacrine ligands? What behaviors can we get out of that? What structures can we build out of that? What immune cell behaviors can we program out of that? So to summarize this talk overall, I want to highlight that computation models are powerful tools for synthetic biology. Simple models like the one I've shown here I made, they work pretty well already for what we have, mainly because in synthetic biology, we program cells and we just have to implement the similar program into our in silico and in vitro system. So with that, that's pretty much the end of my talk and what I presented so far. We can open it up for questions. So I'll open up a simulation. I'll show how it's run. This is the GitHub page, and Hayden's already put it on the Google Drive, so you can go ahead and ignore this GitHub link here. But in the future, I'm going to keep this link here. It's going to have the repository to the codes. So if you guys want to, you can go up and download the codes anytime, run them. I want to emphasize for this type of research, I'm an independent researcher. It's my personal belief that for these types of work, these simulation works, is that you have fun. Don't be afraid to change stuff up. See what happens. Change things. See what happens to the behavior. Find something interesting. And that's kind of how I approach this computational work here. It's interesting because you can change a few parameters, and you can, from just changing a few parameters, you can totally change the behavior of your system. So that's one of the attitudes that I like to take towards this work. I'm going to run these simulations in just a bit. I want to highlight that these simulations unfortunately take 2 to 3 hours to run on 4 threads, which is what I've coded for default. So you need to check your computer system and see how many threads your system is capable of handling. If your system is running a CPU, for example, that has 4 threads maximum, don't run it at 4 threads. It's going to try to take all 4 threads. You should lower it to 2. And I'll show that parameter as I open up the code as well. So instead, in this Dropbox link, what I've posted is the lattice files. And these lattice files for the microscopists in our room, they're effectively the Z-stacks of our structures. But unlike a selective Z-stack where you only take a small section of it, this is a Z-stack of your entire structure at any given time point. So you can literally run the simulation, open up these lattice files, and look at the development of the structure on all slices and from a 3D perspective over the course of your simulation. I'm going to show that too in this demo here. So with that, I'm going to go ahead, close out of my presentation, and I'll just show how to run the code shortly. And this is a good time. We can also have questions asked as well. Let me stop sharing first so I can close my. There we go. I'll minimize this.
Any questions for Calvin about what we've seen so far?
That's taking a little time to open.
Yeah, feel free to unmute
ask me
and ask.
questions.
Maybe I have a question. Your gray cells, they don't
Yes.
have any receptor, and so they cannot sense the other cells, but still they stick to your sphere. How is this encoded?
Oh, sorry about that. So the gray cells, let me go back to. So the gray cells actually do have I just didn't draw that in the signaling network because the synthetic juxtacrine receptor is constitutively expressed. Here you go. Let me share the screen for the slide again. So in our signaling network, I just didn't really show I didn't show too much of the synthetic juxtacrine receptor part. That part's hard-coded, not hard-coded, but rather we built that into our equation for how they actually sense the cells. So our gray cells do have a synthetic juxtacrine receptor that senses the triangle ligand that's present on the blue cells. And yeah, and they always have this constitutively expressed. So we don't actually have to monitor the levels of these. In this model, I assume that the receptor is in excess, so this is not a problem. The limiting rate is due to the ligand. But if you encounter a situation where you want your synthetic juxtacrine receptor regulated by another one, or even change, for example, it's sensitive to doxycycline and induction or so forth, you can actually model this directly too. In this model, we assumed there's an excess, but if you want to, you can actually model limiting levels of receptor. And that can make a really cool network, because even if this is low enough or high enough, you can totally change the behavior of the signaling network here. because cells will change their sensitivity naturally, independent of the ligand levels on these two cells. So let me.
And in your in vitro model, the gray cells are the tumor cells, right? They attract the green cells, and the green cells tell the red cells, and the blue cells will kill the tumor cell.
So, are you referring to? Hang on a second. I'll keep this shared for now. Are you referring to the developmental structure, or are you referring to the tumor model?
I'm referring to the real world, where we were. I mean, you told me that you work on T cards, and when I apply this knowledge to this picture, I would say the blue cells are the T card cells.
So I see. So I'm sorry about that. I just reused colors for the tumor setup. There's actually, this is not a tumor. This is just building the 3-layer structure. So there's no tumor killing in this setup here. This is just building the 3-layer structure here. This is totally synthetic development. And I can go back to the tumor setup. See, that's the problem when you start running out of colors. But when you make figures, you want them to have really good contrast because you want it to stand out when you make a figure. But at the same time, you start running out of colors at some point, especially when you have like 10 cells and so forth, and the contrast just becomes bad. But here in our tumor setup, we can effectively program just gray cells with this network here. So this is not for building a 3-layered structure. These are just our synthetic biology CAR T cells with our anti-yellow antigen as the juxtacrine receptor. So when we mix all the 4 of these cell types together, what should happen is that these gray cells will sense the yellow antigen on these dual antigen cells, dual antigen positive tumor cells. So these are tumor cells for our case here. They express the chimeric antigen receptor against your red antigen, and these cells are able to kill both this one and this one. When you perform the control experiments, for example, if you seed only this, and this, you should not see any tumor cell killing because without the first yellow antigen, there's no CAR expression on the surface. So it's equivalent to dysfunctional T cells. If you were to run the tumor of just this one, this one, this one, you need this cell in order for your system to actually work.
Okay, thank you.
Yeah. Let me share TW edit plus. Okay, so I've opened up the file for the 3-layered structure here. There we go. There we go. So earlier I mentioned one of the things that to watch out for when you run these demos is the number of processors that you're using. That's specified in this line right here. I have a 4-core CPU that hyper-thread, so I have 8 threads in total. That's why I'm able to allocate 4 threads to this simulation. In different systems, this will change. For example, at home, I have a server set up, and I can actually run this up to 32 without any issues on these simulations. It's important to note that in the developer manual, when you play with this number here, you can't set too much. Otherwise, I believe it's called race conditioning, where your CPUs try to alter the same quadrant within one another. So what I find is that in general, when I split them evenly, like 4 or 16, so forth, that works pretty well. The key limitation for most desktop CPUs will be the number of actual threads you have available. If, for example, you have a 16-core CPU, should be hyper-threaded to 32 threads. That means you can probably run up to 16 safely, but you can't use 32 because all your threads will be used, and your computer needs threads to keep itself running in the background. So keep this number, if you're unsure, you can keep this number low just to keep it safe, and it'll run slower, but the trade-off is that it'll be stable on your computer. Okay, cool. So that was the part that I want to talk about. This, and yeah, we'll just run a simulation, and we'll start it just to show how it initializes everything. And because those are, the last files are actually one of the most useful tools I find. Again, they're equivalent to Z-stacks that you would do. So basically it's like microscopy, but the program saves all your Z-stacks for you. So you can just run through them as much as you want. So I'm opening this up here. And when I open it up, there we go. Minimize this. We have our XY slice. So to show 3D first, I'll pause our simulation. Our blob initializer initializes them already as a kind of a blob in the center. If you want to change your initial condition, you can change it to a rectangle and so forth, etc. You can use the uniform initializer steppable. We have our XY slice, and the way I like to think about these slices is that it's literally using a microscope. So our XY, this is our focal plane. We're centered at the lattice, we're centered at XY equals 50. Now we can move along the Z-axis. If we click this, you can move along it. You can see we see different focal planes.
Calvin, maybe I'm the only one, but I still see your threaded window.
Thanks for letting me know. Let me share that. It opened up on a different. There we go. Is that good?
Yeah, now we see the player.
Awesome.
At least I see the player. I don't know about the others. Okay.
Thanks for letting me know. So let me put this back at 50. So the way I think about this is I like to think about it as a microscope slices. We're on our XY axes, or sorry, our XY plane. You can see XY here. So basically when we have our structure, we're moving up and down through the Z axis here. So for example, if you have a 3D structure that you're imaging with a microscope, you can see it floating around. You can image different sections. That's exactly what we see here. So I'm going to scroll through the structure. We can see that as we go through it, we can see different focal planes of our structure. Once we're out of that focal plane, or sorry, once we're out of the structure, we get nothing at all. Scroll in the other direction, we see our focal plane for our other parts of our structure, and boom, we run out of it. But this is how the simulation generally looks when it runs, and you'll be able to see cells as you run it. Cells will slowly transform. In the original code, which is kept on CompuCell3D version 3.7.8, I've added some quantifications. I haven't ported those yet. If you're interested in those, you can talk with me. I can help you out with that. Basically, you run the simulation, and one of the tricks to having your simulation run faster is you not close it, but minimize it, and that actually speeds it up because now your computer doesn't have to render the actual simulation. So it's a little bit faster compared to if we open this up. Actually, you probably can't see that because the screen share is only on that thread. Okay. But anyways, if you minimize your player screen, it actually runs a little bit faster. So what I'm going to do now is I'm actually going to open up a lattice file, and I'm going to show. I'm going to stop the simulation real fast. I'll share this. And I'm going to open up a lattice file, which effectively contains all our Z stacks. So I'm going to go to my drops. Comments in the meantime. Cool. And I'm going to open up the output. So this is one example of an output folder that generates. You can see that I saved a bunch of screenshots at the 2-dimension at the XY plane. This is where our lattice data is stored, and this is actually the important part. So you have a step file here that basically initializes your lattice, and it initializes CompuCell3D player for looking at your lattice files or your Z stack. So I'm gonna have that start up. It takes a little bit of time. There we go. There we go. So we can see on the, are you able to see my player screen right now? Is that working?
Yeah, it looks good.
Yeah.
Awesome. Thank you. So this is our time point t equals 0. We see we have our XY. We have our, wow, that's weird. Why is it popping up there? Right. So we have our structure here. This is our structure. At time step 500 Monte Carlo steps, we see that we actually have our blobs start converging onto one another, forming a spherical, more globular structure, I suppose. More spherical blob, I guess is a better answer. But you can see at 3,500 time steps, we converge well to that. I'm just going to show the XY plane. How does it look if you were to look at the slices of the structure instead? You see the structure is not very sorted. It's pretty mixed. There's blue cells interdispersed all through it. And that's fully expected because these cells don't really have differential adhesion to one another. They shouldn't sort. They should actually form a random structure. And let's move over to around 6,000 time steps. And we can actually see green cells start activating around this time step here. And in fact, they're starting to try to form a core of one another. So we scroll through, we see that green cells are activating, but they haven't had a chance to sort it yet. They actually just started activating here for a 3-layer structure. So now we're forming some green cells, but the green cells are activating sparsely, and they're trying to now actually sort to one another. If I advance through a few more time points, I can see by 7,500 time steps, around 7.5 hours, the core should actually be relatively well formed and stable. So if I run through this, we can see the end of our blue shell here. As we go back into the structure, we see our blue shell, and we see our green core forming all about in the middle. Note that we still have some gray cells that haven't inactivated, and we actually have some green cells on the outside. And that's perfectly normal because biological systems are stochastic. This is stochastic. They don't perfectly form a three-layer structure every time. In fact, one of the things that you can look at is when you do the in vitro experiment, you'll find that there's a distribution in the number of three-layered structures that form. Some of them have two cores, some of them have one core, some of them are a complete mess and have like multiple little cores and so forth. And that's just biological variation. That's normal. If you run the simulation enough, you'll see all those structures as well. So let's advance a little bit more. 10,000, 10 hours-ish. We don't have any red cells yet. We have a gray cell in the center that's deactivated, but we don't have any red cells at all. Let's advance a little bit more, 12,000. We actually see our cells starting to deactivate, and this is something we see in vitro. This makes perfect sense because when your green cells are isolated from your blue cells, they don't receive any signal, so they should be deactivating. And what we see here is that the core actually has a little bit of deactivation. These innermost cells that lost contact don't remain green because they don't have contact with blue cells. Let's advance a little bit more. Show the red cells. That's to 16,000. We know at 20,000 if we should already have a formed three-layer structure because that's what we see in vitro. So finally here, we actually start seeing some red cell activation. Again, I'm gonna scroll through the structure as if we were looking at a microscopy Z-stack. We see the outermost layer of our structure. We see it red, we see our core, we see the red layers forming around the core and so forth. And we see it forming our three-layer structure effectively. Now let's go to the 3D version. Let's take a look how it looks in 3D. In 3D, we see our blue shell surrounding our red cells, and inside we can see peaks of the green core. Let me see if I can move this toolbar real fast. There we go. One of the tools that I like to use in CompuCell 3D is if you want to visualize a specific field. For example, if you were looking only at if you're only looking at the green channel for your simulation, or for your in vitro experiment and simulation, what you can do is you can change some of the cell visibility. So my green cells here are type 2. So I'm gonna make cells of type 1 invisible, type 3 invisible, and type 4 invisible. And what that should do is change it to that should visualize only my green cells. So, oh, I didn't apply it, that's why. That's odd. Where's the apply setting?
Should be in the bottom right. For some reason, it looks like it's not drawing the bottom of your window.
Yeah, that's weird.
I wonder if you have to pull it down far enough, like if the window is too small and it's not scaling.
Maybe I can't really maximize. I was hoping that would help fix it too. So I can scroll on this? Doesn't seem to be the case. That's weird.
Can you pull it down any further?
You mean like fully stretch it all the way to the bottom?
Yeah.
Yeah.
Okay, so this might be a bug.
Something else that we found that we can update for the next version. But in general, there's an apply button here, and you can change your cell types to be invisible. For example, if you want to only look at the green color, the green channel, the green cells, you can put in cells of type 1, 3, and 4. So only your cells of type 2 are shown, and you can actually see just the green core by itself. And again, this is very analogous to what you would do on a microscope. You have your different lasers active. You can turn off lasers to only see specific colors and so forth. I guess from the slices. But over here, yeah, we see that we're forming a three-layer structured cool. I ran this for 50,000 time steps, just my default. But let's look at 20,000, which should match our 20-hour time point. And we see that on our 20-hour time point, we get a three-layer structure that looks pretty similar qualitatively to what we would expect in vitro. We have a sorry, I should scroll too fast. We have a blue shell, then we have our red layer, and once we go deeper into our structure, we see our green core. We see our green core forming. We see some green cells that deactivated, and we actually see like a little gray core instead, almost like a four-layer structure. And this is something that we see biologically too. As we continue scrolling through the structure, we see our red shell. Our core gets smaller because now we're going more towards the top of the structure, and we have our red layer, blue shell, and that basically is our three-layer structure here. So I will keep that Dropbox link up for quite a while. If you guys, when you guys have the time, and if you're able to get the same for the lattice for the invisible cells to work, hopefully on your CompuCell 3D, you'll be able to actually play around. You'll look through the outputs of these simulations, so you don't have to wait 2-3 hours to run these simulations. It takes a bit of time because this is a 3-dimensional simulation, and there's usually a fair bit of cells. Oh, blue. When we're in last place, though.
Never mind. That was a stupid joke.
Oh, oh, I get it. Mario Kart.
Okay.
I get it. I was like, what? So we have a few minutes. That talk took a little bit longer than I expected. I'll be like 30, 40 minutes. If you have questions, this is perfect time. Feel free to ask me. In the GitHub link, I have linked to some of my works that relate to this type of, I have links that relate to these type of simulations and different repositories with the codes for those simulations. If you have questions, my email's on there. You can feel free to contact me with questions anytime. I'll do my best to be honest. If you're interested in talking about synthetic biology, let me know. I'm happy to talk about that stuff as well.
All right, so we've got about 7 minutes or so for questions if anybody wants to ask anything. So I'm glad that you showed this lattice file stuff, because this isn't something that we really had planned on talking about this week, and something that I actually didn't know too much about. So it's a super useful feature. I'm gonna have to go back and review this.
Yeah. No, yeah, these are fantastic. I actually exclusively output in the lattice files, because what I like to do is when I generate my 3D structures, I actually don't use the default outputs. What I do is I actually have a code that erases half of the lattice, and I get a nice cross-sectional 3D structure that way. It looks really cool for papers and for movies and stuff.
Mm-hmm.
Yeah.
Actually, this is really cool. So maybe if Hayden has some connection to the programmer, so a slider for both the stack, so the Z stack and the steps would be unbelievably useful for all of us, I guess. And Calvin, can you please put the link of your Dropbox into the chat, please?
Fantastic. Great idea. That's something I should have thought of. I'll add that on right now. There you go.
Thank you.
Yep.
Yeah, and Calvin's slides are in the student materials folder for today as well. So you can get the link from there after this meeting.
I was trained as a biologist, so that's why I keep thinking of these as Z-stacks, because that's what every PI asks for when you do imaging. It's like, you want to get some images? Great. Can you do a few Z-stacks so we can do like colocalization, we do quantifications and all that stuff. We can show through cool 3D pictures at the end of the. That's why. So every time I think about this, like I always want my output like in a lattice format.
And would it be possible to trace back? So when you have your image, let's say you have such a spheroid or something like that, no? For a spheroid outgrow, for example. So you have pictures of a spheroid outgrow in a confrontation assay, where you have the blue cells, for example, to being tip cells, and the red cells being the stalk cells. And you have it at the very end, and you then make a Z-stack, Z-stack. How do you say it? Z-stack.
Yep,
and
I call it Z-stacks, yeah.
And then you have the last round of your Monte Carlo simulation, so to say, in your microscopic picture. Can you trace this back to or via this simulation in the other way around, when you have the blue and the red cells, you put them together in this confrontation assay, and after a while, all the blue cells are outside and build the tip cells. Would this be possible?
It is possible to visualize. It is possible to visualize that. So let's.
So to say, from the last step, the ordered step, to the first step where all cells are mixed.
Mm-hmm. Can you trace where the cell came from, basically? Is that correct?
Yes.
That's a very, very good question. There is a way to trace that. And I've done it before, now that I think about it, actually. So remember that we can give each cell a dictionary. We can assign them a dictionary, and that assigns them a list of values that we can put on. We can give a cell a list of values. You can change that cell's color. So it's, for example, yellow in these simulations. So you specify a specific type, but have that type color behave the exact same as its normal type. So I would say, for example, you want to track this blue cell that goes out to the outside. You can specify a type ID number because the blue cells will have an ID assigned to them. So for type cell ID number 3, check if it's a blue cell. If it's a blue cell, mark it as a type 5. I haven't even specified 5 types of cell, but you can mark it as type 5. This is your pseudo blue cell here, and you can change its color to yellow when you visualize it. So because it has that special ID on it, you can actually see that cell as the yellow cell move throughout your simulation. You can see it move from the center. You can see it move all the way to the outside. Now, the complicated part about that is it increases complexity because now you have to specify a cell type number 5 specifically for that tracking purpose. So if you wanted to track like 5 cells, you have to add almost 5 cells for single cell tracing. And that's going to make your that's extra programming work. It wouldn't add on a computational load, but basically you're adding on a special tracer onto it that you can tell what cell it is, and it's absolutely doable.
Interesting, very interesting.
Yeah, that's the way I've done it in the past. In this new version of CompuCell 3D, there might be a better, more efficient way to do it. That's how I've done it in the past. Because what I did was I used to specify like 4 random cells, and then I would specify them to have a unique cell type that I could track. That cell type, sorry. I would add a cell type 5. I would change that to yellow color and put a W as the name for it. And these cells will behave exactly like blue cells. So wherever there's blue cell, they send signals, receive signal, and so forth. I would just add cell type 5 there, and they behave exactly like blue cells. Only difference is now they have a tracker added to them. So we know that they're where they are at any given time point. And yeah, you can monitor that throughout your simulation. You can see their color is yellow, so you can see where they move at each time point. You can follow them through the slices and everything.
When we have them in a UMAP, for example, or in a PCA, we also can have a pseudo-time,
Yep.
and this would be also very helpful in tracing back, right?
Yes, and tracing where your cells come from. Yep.
Yeah, thank you.
Yeah, of course. Thanks for asking. That's the best way I can think of it right now. There might be better ways in this new version of CompuCell 3D for doing single cell tracing. That's a very cool idea. I like that a lot. This is one of the funny parts here, because in vitro that's actually the easy way, because you can add a cell trace dye to the cells that you want to trace, and you just mix it into it, and that's very easy to trace overall.
Yeah, so okay, we're officially at 4:15. We can probably stick around here a couple more minutes if anybody had any last minute questions for Calvin. But if you don't have any questions for Calvin, we'll go ahead and say thank you again for coming, and we will now return to the main session, which let me drop the link in the chat here. I can remember where I put it. This is the danger of cleaning up and organizing your files in the middle of a big workshop like this, as you're sense memory is now gone. All right, so here's the Zoom link for the main session for those who'd like to return back now. And we'll start module 5.4 at 4:30 p.m. Eastern time. So we'll see you back there at 4:30.
Thanks for attending, everybody. Appreciate it.
Yeah, like I was saying, this is super cool. I'm gonna have to look into the lattice file stuff because we actually had one of the guest presenters yesterday who wasn't able to. She showed her code, but she wasn't able to like have people walk through a demo. First of all, because her simulation required a bunch of like custom packages that you had to install. It was using a couple of special plugins, and the code was really complicated, but it also ran very slowly. So she was like, even if we were to run it, we'd only be able to see like, the first little bit of it. They won't see the actual interesting part of the model. So we'll have to look into getting people to generate lattice files for us for these kinds of demos. Because I wonder if the lattice file also gets around the plugin problem.
I would think so, because so the lattice files, when I read them you can just read them as a text file.
Yeah, that's
the
what
way
I figured.
I remember that they store very basic parts like the cell.id. They even store the number of protein points that cell. It just stores the ID and the type. So it literally just stores enough to visualize your structure based on their color. So they do add a lot of they do take a lot of space, because if you save very frequently, they take up a lot of space. It's just a lot of text. But this is literally the only way I go about doing CompuCell 3D.
Yeah,
because later on I want to change colors, or I want to change the visualization a little bit, have better contrast. I need to go back to the last files, and I can just change it right there, and it's like I don't have to rerun anything,
Right. Yeah, it's pretty cool. They're basically just like sophisticated bit maps, more or less, that are sequenced. Yeah, I'd heard we talked about them before, but I haven't really looked into them much. So yeah, that's super cool. I'm glad that you introduced those today.
They work pretty well in general. I've never really had an issue with lattice files, honestly. The feature works. It's been great since CompuCell 3D version 3.7. It's been great now.
Yeah.
Hey.
Hasta, do you have a question?
Yeah, it's kind of a general question, actually. First of all, thank you for such a fantastic presentation. I really enjoyed it.
Thank you.
The thing is, my background is also in molecular biology. I have a bachelor's in molecular cell biology, and I'm just starting to learn about biological modeling and also synthetic biology in parallel, of course. So I was wondering if you have any insights on how to go about that, which resources to use, or how did you get started? Like I said, it's a very general question. I apologize.
No, that's a very good question. So I started because I was lucky to work in a Leonardo's Morset lab. He's one of the top synthetic biologists in our field right now. So I was able to get experience through there. I got exposed to a lot of different ideas, and importantly, these synthetic receptors. I can tell you right now that the people whose works you can look at if you're interested in synthetic biology is definitely look up Wendell Lim. He's at UCSF, and actually I will type it into chat so you can just copy it. That'll save you a lot of time.
Oh, thank you so much.
Yeah, of course. He's actually just released a. Oh, go ahead.
Interrupt. My internet connection is a little spotty, so I keep getting lagging in and out.
No worries. Let me know. Let me know if you can see this. Cole Roybal does a synthetic immunotherapy. That's his niche. And Leo. USC. I better specify which USC this is. So if you can see those three names, those are people whose work I would say look into. They also have a lot of collaborations with other synthetic biology labs. So if you look at the last of the people in the author list, chances are those last three authors, you can look at their works as well, and there's a lot of synthetic biology stuff to do there.
Thank you so much.
And if you have more specific questions and stuff, you can shoot me an email in the future. That's no problem.
I really appreciate that. Thank you.
Yeah, no worries. I might have spelled Wendell wrong. Maybe it's an E. Maybe it's a Wendell. but I'm pretty sure he'll pop up in Google anyways.
All right. Well, I think with that, then I probably need to head back to the main session now. So Hasti, yeah, just let me know if if you want to follow up with Calvin at some point, I can put you two in touch.
Of course, thank you.
All right. I'm going to go ahead and stop my recording here.
Same.